This video is about the A-level biology topic of lipids, concentrating on the structure and function of the triglycerides, the phospholipids, and also the steroids. Lipids are a class of biological macromolecules containing the solid fats and the liquid oils. Living organisms make use of lipids for a really wide variety of different functions, starting with energy storage. So lipids are a much more efficient store of energy than carbohydrates. If you took 100 grams of fat and 100 grams of sugar, the 100 grams of fat would have a lot more calories stored in it. And generally speaking, when we're thinking about plants and fish, this tends to be in the form of oils, so that means liquid lipids at room temperature, whereas other animals tend to store these lipids as fats, so solids at room temperature. The second really important function of lipids is within plasma membranes, both around cells and also around certain subcellular structures or organelles. And there are two main components here that contain lipids. One is the phospholipid bilayer, so this makes up the vast majority of that cell membrane. And then the second thing is the cholesterol. And you might think that cholesterol is a bad thing, but actually all of your cells need some cholesterol in the cell membrane, and this helps to moderate the fluidity of that membrane. The third thing that we use lipids for is thermal insulation. So on the previous slide, I had a picture of a seal with all that blubber that makes it very fat, but it's also there to make sure that it's not getting too cold. So we particularly see this in marine mammals that live in very cold environments where they have a layer of subcutaneous fat which helps to insulate them against heat loss. But that fat also has another function. As well as it keeping those mammals warm, it also aids them with buoyancy, so it makes it easier for them to float, because as we know, fat is less dense than water. The lipid can also act for a bit of protection because it acts as a shock absorber, and also lipids can be modified to be turned into hormones. So we're going to look at a few different examples of hormones that are based on lipids. Some vitamins are fat soluble and therefore you need to have lipids as a solvent, and then also in order for your nerves to function properly and for the nervous impulses to move quickly down a neuron, those nerve cells, those neurons, have a myelin sheath. So this is sort of a coating around the outside of the neuron that allows the electrical impulse to move much more quickly. The first type of lipid we're going to look at is the triglyceride, which is the most common type of lipid. So this is what we find in butter and in olive oil. And the molecule has two different components. So firstly, we have this glycerol molecule, and we'll look in a second at what that actually looks like. And then bonded to that, we have these three fatty acid chains. Now, these are essentially really long hydrocarbon chains with just a modified functional group that's going to help them to bond to the glycerol. So they're usually drawn skeletally because they can be quite long and quite complicated. But if you're not doing A-level chemistry and you're not used to this way of drawing things, basically each one of these vertices, each one of these corners represents a carbon atom, and then coming off that are a load of hydrogen atoms which aren't drawn. But we can simplify this for now and just say we've got our three fatty acids which are bonded to the glycerol. And those fatty acids could be the same as each other, or they could be three different fatty acids. Now, we've said that energy is stored within that lipid. There are two different options for getting that energy out. So one option is that there is energy stored in the bonds between the glycerol and the fatty acid. So when digestive lipases break down the lipid into glycerol and fatty acids, those bonds are being broken and that energy is being released. And then the other option, which we didn't really talk about at GCSE, is that following that hydrolysis, following that breaking apart of the lipid, those fatty acids can be transported to the mitochondria, and the energy that is stored in the bonds of that hydrocarbon tail can also be released by breaking those down. So triglycerides are really commonly found as an energy store in a type of fatty tissue called adipose tissue. And as well, that's one of the places that we're going to find fat-soluble vitamins. Now, as we've said, the first component that makes up a triglyceride is a glycerol molecule, which looks like this. Now, if you're doing A-level chemistry, you might also want to know what the systematic name of this would be. So this is propan 1,2,3 triol, but for the purposes of A-level biology, we're just going to refer to it as glycerol. And then bonded onto that glycerol, we're going to have these three fatty acid chains. So to start drawing a fatty acid, you're going to start off with a long chain of carbons. and then around most of those we're going to add hydrogens to just make basically an alkane. But you can see we've got one carbon left on the end which does not have hydrogen surrounding that, and that's going to be part of a carboxyl group. So this is the functional group of a carboxylic acid if you took GCSE chemistry, triple science, or if you're taking A-level chemistry now. And what's going to happen is that when the fatty acid joins together with the glycerol, that acid group is going to react with the OH groups on the side of the glycerol there. Now, as we said, usually fatty acids, because they are quite long, are drawn skeletally, because this example that I've drawn here is actually way smaller than a fatty acid would be, but I'm just keeping things a little bit simple. So your skeletal drawing, we would still draw the functional group of the carboxyl group at the end, but then as you can see, we basically use this sort of zigzag line, and everywhere that it changes direction, everywhere that you have a vertex, that represents a carbon. Now, one of the things that we said at the very start of this topic is that lipids are not polymers. They are macromolecules, they are very large molecules, but they're not polymers. So pause the video and make sure that you can explain why a lipid is not a polymer. Lipids are not polymers, and there are two key reasons for that. One is that they aren't a very, very long chain, and the other thing is that they're not made of repeating units. So in DNA, we have nucleic acid after nucleic acid after nucleic acid. In a polysaccharide like starch, we have glucose, 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 glucose. But in a lipid, we have a couple of different components that are joined together. We don't have one component being repeated over and over. In the carbohydrate topic, we already met the idea of condensation reactions, and these are important for lipids as well, because this same type of reaction is going to be responsible for joining our fatty acids to our glycerol molecule. So a condensation reaction is where two smaller subunits join together to form a larger molecule with the loss of a small molecule, and in all of these biological examples, that small molecule is going to be water. So we start off with our glycerol molecule on the left-hand side and the fatty acid to the right. Obviously, for a triglyceride, there are going to be 3 of these fatty acid molecules, but I'm just showing you one to keep it straightforward. So the carboxyl group of the fatty acid and the hydroxy group of the glycerol are going to bond together with the loss of this water molecule. And that's going to produce this much larger molecule, and you can see here that we have in red the ester bond. So you need to be aware that in a triglyceride or in any lipid, the glycerol and the fatty acid are held together by an ester bond, which has this functional group, which is a carbon double bonded to an oxygen and single bonded to another oxygen. So overall, our triglyceride, if we drew it out properly and drew all of the atoms involved, would look something like this. In this instance, I've drawn you 3 identical fatty acids, but as we've said, the fatty acids don't need to be the same as each other. Time for a quick progress check. So pause the video and make sure that you can draw glycerol, draw some fatty acids, join these together by a condensation reaction, and identify the type of bond. So your glycerol molecule should look something like this. Your fatty acids all need to have the carboxylic acid group on the end, the carboxyl group, and then some kind of zigzag to represent that hydrocarbon chain, but the length is not going to matter. And then when you join them together, we're going to lose a water molecule in each instance and join them up, and this type of bond is an ester bond. You've probably heard of saturated and unsaturated fats in the context of what you eat, but what actually is the difference and where do we find them? Well, generally speaking, saturated fats tend to be animal fats, and these tend to be solids at room temperature, whereas unsaturated fats tend to be things like olive oil or vegetable oil that have come from plants, and they tend to be liquids at room temperature. You learned in GCSE chemistry that a saturated hydrocarbon has every carbon bonded to the maximum number of hydrogen atoms, and therefore it only contains single covalent bonds, whereas an unsaturated hydrocarbon, like an alkene, contains some double bonds, because not every carbon is bonded to the maximum number of hydrogen atoms. So in the context of fatty acids, we can use the same language to talk about whether there are any double bonds in that hydrocarbon chain that makes up part of the fatty acid. So this would be a saturated example of a fatty acid because it doesn't contain any double bonds, whereas unsaturated fatty acids don't have the maximum number of hydrogen atoms, and therefore they do contain carbon-carbon double bonds. So now that you know the difference between a saturated and unsaturated fatty acid, Pause the video and draw one example of each. So it's up to you whether you've chosen to draw the skeletal formula or a full displayed formula, but either way your saturated fatty acid should not contain any double bonds, and it's important that you have drawn out the carboxyl group, so that carbon double bonded to oxygen, single bonded to another oxygen which is bonded to hydrogen. And then for your unsaturated fatty acid, again you could choose to draw it as the skeletal formula or the full displayed formula, but if it's unsaturated it needs to contain those double bonds. Just like we saw with carbohydrates, where we use condensation reactions to make disaccharides and polysaccharides, and then hydrolysis to turn them back into monosaccharides, fatty acids and glycerol can be split apart from a triglyceride using hydrolysis reactions. So instead of taking a water molecule away, we're now adding one back to get those original components. Here is a really simplified diagram of a triglyceride. So now that you know what hydrolysis is, can you draw what the products will be if this triglyceride undergoes complete hydrolysis? In other words, it's been hydrolyzed as far as it can be. So with a bit of luck, you managed to draw a glycerol molecule, and then you've drawn some fatty acids that have a carboxyl group on the end, and there are 3 of them. So why does that saturatedness or unsaturatedness matter, and how does this have an impact on whether the lipid is a fat or an oil? Well, as you can see here in this diagram, where you have that unsaturatedness, that double bond, this causes the fatty acid chain to kind of kink, and this is going to mean that those fatty acid chains are unable to pack together as closely. So whereas you can pack quite a lot of saturated triglycerides into the same space, unsaturated triglycerides are not going to pack together as closely, and this is going to mean that the weakened molecular forces between them are less strong, and therefore it's easier to melt them. We can also categorise unsaturated lipids further based on whether they have just one double bond and are monounsaturated, or whether they have many double bonds and are polyunsaturated. Pause the video while you recap what we've covered so far. Lipids contain carbon, hydrogen and oxygen. Triglycerides are made from a glycerol molecule joined to 3 fatty acids. These are joined together by ester bonds. Unlike a saturated fatty acid, an unsaturated fatty acid contains one or more carbon-carbon double bonds as part of the hydrocarbon chain. Which of these 4 options best describes this triglyceride? We can immediately rule out option number 4 because peptide bonds are found in proteins in between the amino acids. The bonds in this molecule are ester bonds. So now we need to decide, is this triglyceride saturated or unsaturated? And if it's unsaturated, is it polyunsaturated or monounsaturated? We can see that one of the fatty acid chains in this triglyceride is a saturated fatty acid chain, but we're thinking about the triglyceride as a whole. So we need to look at the number of double bonds. Poly means many, and here we do indeed have many double bonds, 4 in fact, so our correct answer is going to be B. Phospholipids are modified triglycerides that are absolutely vital for the formation of membranes in cells. In addition to the glycerol and fatty acid molecules that we're already familiar with, they contain an inorganic phosphate ion in place of one of the fatty acid chains. The basic phosphate group is PO4 3-, although there may be additional R groups. The phosphate group and the glycerol molecule together make up a slightly charged head. Due to the slight charge, this head is hydrophilic. In other words, it's attracted to water. The fatty acid tails, on the other hand, are uncharged. Remember, they're just hydrocarbons. Therefore, they are slightly hydrophobic and they repel water. Typically, phospholipids have one saturated and one monounsaturated fatty acid chain in order to maintain fluidity of the cell membrane by stopping the phospholipids from packing too closely together. However, organisms that live in colder environments are more likely to have higher levels of unsaturatedness to maintain membrane fluidity at lower temperatures. Phospholipids are amphipathic, and this is really important because the fact that they have a hydrophobic end and a hydrophilic end is vital for understanding why they form bilayers and micelles. So when you put phospholipids into water or into an aqueous solution, one part of the molecule is being attracted to the water while the other part of the molecule is being repelled. And so when you have multiple phospholipid molecules, they're going to orient themselves in a way where their hydrophobic parts are clustered together and are away from the water. And depending on the length of the fatty acid chains and how much of this phospholipid you have, they will either form a bilayer or a micelle. The phospholipid bilayer is the basic structure that all of the cell membranes are based on. So we have extracellular water outside the cell, we have intracellular water inside the cell in the cytoplasm, and between these we have this double layer, where both halves of the layer have their hydrophilic head touching the water and their hydrophobic tail away from the water. A micelle is a much smaller structure, and the reason that most phospholipids don't form a micelle is just because their fatty acid chains are too long, and so if they tried to form this kind of structure then they would interfere with each other. But if you have phospholipids with quite short fatty acid chains, then instead of forming a long flat bilayer, it's possible for it to form this smaller structure where it's just a single layer with all of the hydrophobic tails inside and the hydrophilic heads outside. If we're going to be specific about which part of the water the phospholipid is being attracted to, remember that the phospholipid has a slightly negative charge, and therefore it's going to be attracted to the slightly positive part of the water, which is going to be the hydrogen atoms. Some phospholipids are modified even further by the addition of one or more carbohydrate chains. These specialised phospholipids are called glycolipids, so remember glyco is anything to do with sugars. These glycolipids have roles in maintaining the membrane stability, cell recognition, facilitating cell-to-cell adhesion where cells stick together, and also as receptors for cell signalling. You've probably heard of steroids in the context of anabolic steroids, which are drugs that people take in order to build up muscle mass. But actually, steroids are a much wider class of compounds that have many different uses within living organisms. And they're all based on this basic structure that has 3 6-carbon rings and 1 5-carbon ring. And depending on the different atoms that are bonded to those rings, we can make a really wide variety of different molecules. The first one that you need to know about is cholesterol. Cholesterol is a fundamental building block for cell membranes alongside the phospholipids that we already know about, but also it can be modified further to create hormones and also vitamin D. Cholesterol is an ever so slightly polar molecule on one side, but the vast majority of it is nonpolar and hydrophobic. Cholesterol is a small, thin molecule that can go in between the phospholipids in the phospholipid bilayer. Because it has that slightly charged end of the molecule, this can interact with the hydrophilic heads of the phospholipids, whereas the rest of the molecule, which is uncharged, interacts with the hydrophobic tails of the phospholipids. The cholesterol is included in the membrane because it increases the rigidity by packing the phospholipids closer together. We sometimes describe this as regulating the membrane fluidity. With slightly different modifications to our steroid rings, we can make a number of steroid sex hormones such as testosterone, estrogen and progesterone. You can see that all 3 of these sex hormones are based on that original steroid ring, just slightly modified with different atoms. Cholesterol can also be modified to make glucocorticoids like the stress hormone cortisol and also vitamin D. So for a final summary, pause the video and make sure that you can complete some notes about the structure, main role and other features of triglycerides, phospholipids and cholesterol. Hopefully you remembered that while triglycerides are formed from a glycerol molecule and 3 fatty acid chains, in a phospholipid one of those fatty acid chains is replaced by inorganic phosphate. Cholesterol is based on those 4 carbon rings, so 3 6-carbon rings and 1 5-carbon ring joined together. Triglycerides are a really useful compact energy store and they're much more energy dense than carbohydrates are. Phospholipids are a crucial part of the cell membrane, and cholesterol is the basis of the stability of that membrane, but also a number of different steroid hormones. So triglycerides are typically stored in animals as fat and used for thermal insulation and protective properties as well as their buoyancy. Phospholipids can be modified to produce glycolipids, and as we said, cholesterol is the basis of various steroid hormones like the sex hormones as well as cortisol. Thank you very much for watching, and I hope you found that a useful summary of the lipids topic for A-level biology. If you did find the video useful, then don't forget to like, subscribe, comment and share, and I'll be back soon with some more A-level biology resources.